So today we'll see in detail about the gynecium, which is the female reproductive part, and under that megasporangium, that is ovule. Ovule is also called as integumented megasporangium, that is it is surrounded by integument. And then about the embryo sac. So what is an embryo sac and how it is formed, right? So this embryo sac is actually it is a female gametophyte. Pollen grain is a male gametophyte and embryo sac is a female gametophyte. So first we'll see about the in general about the gynecium, which is a female reproductive part. So which is the female reproductive part? It is carpels or the pistils, right? Carpels and pistils. So if you take the female reproductive part, that is the gynecium, it may have a single pistil. That is, it is either a monocarpillary or it may have more than one pistil. So single pistil or multiple pistil. If it has only one pistil, it is monocarpillary. And if it is more than, if it is having more than one pistil, it is multicarpillary. And this carpel is also called as megasporophyll. Megasporophyll. Whereas stamen is called microsporophyll. Stamen is called microsporophyll and carpel is called megasporophyll. Okay. So in multicarpillary, the pistils may be either fused together. So all these terms actually we already saw under the angiospermic plants, right? So I hope you are familiar with these terms. What is syncarpus, apocarpus, everything, right? So the pistils may be fused together, that is syncarpus, or it is free, that is apocarpus. So these are the examples for some of the apocarpus and the syncarpus. So this hibiscus, this is the hibiscus pistil. So all the other parts are removed, that is calyx, corolla, everything is removed, only the pistil is shown. That is, it has the three parts: stigma, style, and then the ovary, which is attached to the thalamus, that is the basal part, right? So only the female reproductive part is shown here of hibiscus. So here it is an syncarpus pistil. And this B is the multicarpillary, that is more than one pistil, and it is a syncarpus pistil. So here also it is a fused pistil of papaver. So example is papaver. So here it is fused together. And this is an example for apocarpus, that is free. Apocarpus gynecium of Michelia. So they may ask you what is the type of the pistil that is present in the Michelia, or what is the type of pistil that is present in papaver or hibiscus. So you be clear with it. Okay. So multicarpillary syncarpus pistil of papaver and multicarpillary, this is the apocarpus gynecium of Michelia. Okay. So these are some of the examples for the syncarpus and apocarpus pistils. So if you take the structure of the pistil, already we saw it as the stigma, style, and the ovary. So stigma is the main part, that is, it receives the pollen grain. So whenever the pollen grains, mostly it is transmitted through the wind, or it may be by some insects or some animals, right? So it is landed on the stigma. So stigma is the landing platform for pollen grains. So this is the stigma, and then style. It is an elongated slender part beneath the stigma, and ovary is the basal bulged part, and inside that ovules are present, right? So you're familiar with all these structures, right? And inside the ovary is the ovarian cavity, that is locules are present. Inside the ovary, there is an ovarian cavity where placenta is located. So inside is the placenta, and even we saw about the different types of placenta, everything, right? So inside the ovary is the ovarian cavity in which placenta is located. And arising from the placenta are the ovules. So from where the ovules arise. So these are the ovules. So arising from the placenta are the ovules, which is also called the megasporangia. So Megasporogenesis is nothing but a ovule. So they are present inside the ovary. Okay. So the number of ovules in an ovary may be either one. In some cases, only one ovule is present. So examples are wheat, paddy, mango, all these are having only one ovule inside the ovary. But in some cases, many ovules are present. Example, papaya, watermelons, we can see many seeds inside, right? So the ovules may be single or it may be many. So be clear with the examples. Okay. So next we'll see about the structure of the ovule, right? So this is the structure of the ovule. So if you see the structure, the ovule is attached to the placenta by a stalk. So where the ovule is located, it is present in the ovary. So it is attached to the placenta by a stalk. So this is the funicle. So this is the stalk, and through this stalk, this ovule is attached to the placenta. Okay, with the help of a stalk that is called the funicle. And junction between the body of the ovule and the funicle is called the hilum. So what is hilum? The junction, because so this is the body of the ovule. All these are the body of the ovule and the funicle. And this body of the ovule is connected to the stalk or the funicle through the hilum. So it connects the body of the ovule and the funicle. So that is called the hilum. Okay. And each ovule has one or two protective envelopes that is called the integuments. So this is an outer integument. So this outer layer is the outer integument and inner is the inner integument. So generally it will have the two protective layers, but in some cases it may differ. Okay. So it has the two protective envelope, that is the outer integument and the inner integument. And the role of this integument is, that is, they encircle the ovule except at a tip. So this integument, it covers, for example, if you see, it covers the entire ovule, right? It is present throughout the ovule, but except at the tip, where a small opening. So this is the opening, right? And it is called the micropyle. So except at the micropylar region. Okay. So each ovule has one or two protective envelopes that is called integuments, and they encircle the ovule except at the tip, where a small opening, that is micropyle, is present. Okay, so this is the micropyle where the opening is present. And opposite to the micropylar end, so this end where the micropyle is present, that is called the micropylar end. So opposite to this micropylar end, so this is the opposite end, right? It is called the chalasa or this chalasal pore. So opposite to the micropylar end is the chalasa, which is the basal part. Okay, so these are the different parts of the ovule. Then enclosed within the integuments, there is a mass of cells called nucellus. So which is nucellus? Here, the center. So enclosed within the integument, so outer integument, inner integument, and within that is the nucellus. So this nucellus, actually, it contains the reserve food materials. Okay, so it has the reserve food materials. So it provides nourishment. So inside the nucellus is the embryo sac. So in the center, this is the embryo sac, which is also called the female gametophyte. Okay, so the center is the embryo sac. So an ovule generally has a single embryo sac. So each ovule will have a single embryo sac. So how the embryo sac is formed? It is formed from a megaspore. So how the megaspore are formed? Through the reduction division, right? So an ovule generally has a single embryo sac which is formed from a megaspore through reduction division. Okay, so I hope you are clear with the structure of ovule, right? So this is the structure of ovule. So now, based on the number of integuments that are present, there are different types of ovules such as unitegmic, Just unitegmic ovule. So what do you mean by unitegmic? Uni means single. So unitegmic is a single integumented ovule. It is a single integumented ovule. So here examples are mainly members of gamopetalae. So gamopetalae and mostly gymnosperms. They will have unitegmic ovule and gymnosperms, right? And there are different types such as bitegmic. So from the term itself, we can say it as two integuments, right? Two integuments. So example is polypetalae. That is most angiosperms. That's what we saw now, right? That is, it has the two integuments, that is outer and the inner integuments. So most of the angiosperms, they have the two integuments, mostly angiosperms, right? So polypetalae and then monocots, mostly they have two integuments. And ategmic. So what do you mean by ategmic? If there is no integument, right? There is no integument or integuments are absent. And in rarely some cases, they are surrounded by three integuments. Some cases, three integuments. So based on the number of integuments that are present, there are different types of ovules. And there are types of ovules which are based on the nucellus that is present. That is nucellus. Nucellus is a part which is present inside the integument, right? And outside the embryo sac. So based on the presence of that is the type of nucellus that is present, it is called tenunucellate. So here the nucellus is less developed. Or it is present in the form of a single layer, tenunucellate, or it may be gracinucellate. So here, massive type of nucellus, that is, it will be present in many layers. Here only less developed or single layer it will be. So here it is more layered. Massive type of nucellus. So here the main examples are monocots and also the polypetalae group. They have the gracinucellate. That is massive type of nucellate and it is present in many layers. Okay. And in some cases, so what do you mean by perisperm? We already saw these terms, right? So in some angiosperms, what happens is some of the nucellus that will remain actually inside the ovules. But in most of the cases, what will happen is the nucellus will be used up. But in some cases, the nucellus will be present, visible. So the part of the nucellus present inside the seed. The part of the nucellus which is present inside the seed in the form of a thin layer. That is called the perisperm. So that is called the perisperm. But in most cases, the nucellus will be used up, but in some cases, the nucellus will be present inside the seed. After the seed is formed, it will be present inside in the form of a thin layer that is called the perisperm. Okay, so these are the different types of ovule that is based on the number of integuments and based on the nucellus that is present. So based on the number of integuments that is present, it is classified into unitegmic, bitegmic, and ategmic. And in some cases, rarely even 3 integuments are present. And based on the type of nucellus, that is tenunucellate and gracinucellate. Tenunucellate is the one in which the nucellus is less developed, In the case of grassy nucellate, it is a massive type of nucellus. Examples are the polypetalae and the most monocots. They have the grassy nucellate type of a nucellus is present. And perisperm is a part of the nucellus is present inside the seed in a form of a thin layer that is called the perisperm. So, for example, is pepper. Perisperm is present. So even after the seed is formed, the nucellus will be present. So nucellus is actually, it is the reserved stored food, right? So, and then based on the type of the position of the micropyle and the stalasa and the hilum. So we saw what is a micropyle, what is a stalasa, and then what is a hilum. So now you're familiar with all these terms, right? And the funiculus, that is the stalk. And then the body of the ovule is connected to the stalk by hilum. And we saw about the micropylar end and the chalosal end, right? So you're familiar with all these things. So based on the position of all these, that is where the micropyle, where the chalosa, hilum, and the funiculus, there are different types of ovules such as orthotropous ovule, anatropous ovule, hemitropous ovule, Amphitropus, and then Camphylotrophus, and then Trophus. So these are the 6 types of ovules which are based on the position of the micropyle, chalosa, hilum, and funiculus. So for example, if you take the orthotropous ovule, the first type is orthotropous. So what is the special feature of the orthotropous ovule? It is called the upright ovule. So if you take the structure here, the micropyle, chalosa, and hilum, everything will be in a straight line. So this is an orthotropous ovule. So all the position of micropyle, chalosa, and hilum will be in a straight line. And this is the most primitive type. Most primitive and simplest type. And simplest type of ovule. Okay, and next type is Anatropus. So what about the Anatropus? First, Orthotropus, Anatropus. So here it is inverted. When compared to this structure is an inverted. So it is called an inverted ovule. Inverted ovule. And mostly 80% it is present in 80% number of angiosperms. Anatropous ovule is present in 80% families of angiosperms. Okay, so actually it is an inverted ovule. So here the micropyle and chalosa are in the opposite end, but hilum is, it's nearer to the micropyle, right? So here the micropyle is facing upwards, but in the case of Anatropous ovule, the micropyle is facing downwards. So it is an inverted ovule. And the next type is Camphylotrophus. Camphylotrophus. So it is also called curved ovule. Camphylotrophus is a curved ovule. So if you see the structure, this is the Camphylotrophus. So they are not present in a straight line. Micropyle and chalosa are not present in a straight line, and it is a curved ovule. That is the Camphylotrophus ovule. And the next type is Hemitropus. Hemitropus ovule, which is also called horizontal ovule. So actually, this is the structure of Hemitropus, and this is in between Orthotropus and Anatropus. So here it is an upright, and this is an inverted, and the in between is the Hemitropus ovule, that is at the 90-degree angle, right? So micropyle and chalosa they are in a straight line, but hilum is not in a straight line, and this is an horizontal ovule. So it is also called horizontal ovule. Okay. And next is the Amphitropus. So here, if you see the structure, this is the structure of Amphitropus. Here, this is the embryo sac. The embryo sac is in the form of a horseshoe. Embryo sac is horseshoe shaped. And it is also called transverse ovule. Transverse ovule. And then last one is the Trophus. Trophus ovule. So this is also called coiled ovule. So if you see the structure of this Trophus, this tunicle actually it surrounds the entire ovule. So this is the micropyle and then chalica. This tunicle, if you see, it surrounds the entire ovule. So it is called the coiled ovule. Okay, so it is surrounded by funiculus. So based on the type or the position of the micropyle, chalica, hilum, and the funiculus, there are 6 different types of ovules. That is Orthotrophus, Anatrophus, Camphylotrophus, Hemitrophus, Amphitropus, and Trophus ovule. So Orthotrophus ovule is also called upright ovule. That is, it is the most primitive and simplest type. All the micropyle, chalica, and hilum will be in a straight line. So it is called the upright ovule. Right, and Anatrophus ovule is called the inverted ovule. The micropyle will be facing downwards, and this type of ovule, that is Anatrophus ovule, is present in 80% of families of angiosperms. And third is the Camphylotrophus, and it is called the curved ovule, and Hemitrophus is also called the horizontal ovule, and Amphitropus. So Hemitrophus is in between Orthotrophus and Anatrophus ovule. And Amphitropus ovule, here the embryo shape is horseshoe shape in the case of Amphitropus ovule, and it is called transverse ovule. And then Trophus ovule, example, that is, it is also called coiled ovule, and the funiculus, it surrounds the entire ovule. So these are the different types of ovules. Okay, so based on the position of micropyle, chalica, hilum, and funiculus. So clear? So we saw about the different types of ovules on the basis of number of integuments, and also the type of ovule based on the type of nucellus that is present. So this is based on the position, right? Then what is Megasporogenesis? So Megasporogenesis means formation. So how the megaspores are formed? So it is the formation of megaspores from megaspore mother cells. Right? So how the megaspore mother cells, they form the megaspores. So here the ovules, they differentiate a single, MMC means it is a megaspore mother cell, right? That is, they differentiate a single megaspore mother cell in the micropylar region of the nucellus. So now you are clear with the different regions, right? Micropylar region and then chalical region, and you know clearly about the structure of the ovule. So here the ovule differentiate a single megaspore mother cell in the micropylar region of the nucellus. So this is the nucellus and the micropylar end. So this is the micropylar end of the nucellus, and this is the megaspore mother cell. So this cell, actually it is a large cell containing dense cytoplasm and a prominent nucleus. So it has a prominent nucleus, and this megaspore mother cell, the megaspore mother cells, it undergoes meiosis, right? And it undergoes meiosis. First, it forms a megaspore dyad, that is, it divides into 2 cells, and then megaspore tetrads. So it undergoes meiosis to form 4 megaspores. So 4 megaspores are formed. Right? So this is the process of megasporogenesis. So the formation of megaspores from megaspore mother cell. Okay. So the ovules, they differentiate a single megaspore mother cell in the micropylar region of the nucellus, and it undergoes meiosis to form 4 megaspores. So in the majority of the flowering plants, we saw that 4 megaspores are formed. So what will happen to the 4 megaspores? Only one is functional, out of the 4 megaspores, only 1 megaspore is functional, while the other 3 degenerate. So only 1 megaspore further develops into a female gametophyte, right? So the functional megaspore, it develops into a female gametophyte. So the embryo sac formation from a single megaspore is called the monosporic development. So this term is more important. Why it is called monosporic development? Because though 4 megaspores are formed, only 1 megaspore is involved in the formation of embryo sac, right? So it is called the monosporic development. Okay, clear? So any doubts till now? Are you clear with this about the structure of ovule and also about the process of megasporogenesis? Clear, right? Yeah. So it is called monosporic development. So now how the embryo sac is formed? The nucleus of the functional megaspore, that is the nucleus of the functional megaspore. Functional megaspore means there is the 1 megaspore that develops into one embryo sac. So the nucleus of functional megaspore divides mitotically. So that nucleus, it divides mitotically to form 2 nuclei. So this is the first stage. So this is the 2 nucleate embryo sac. So first stage is the nucleus of the functional megaspore. It divides into 2 nuclei, and then this 2 nuclei, it moves to opposite pole. That is one nuclei at the one end and the other nuclei at the opposite end. So they form an 2 nucleate embryo sac. So this is the 2 nucleate embryo sac, and then it again divides to form an 4 nucleate. So this is the 4 nucleate. So mitotic division happens and it forms a 4 nucleate embryo sac. And then again, third mitotic division occurs to form an 8 nucleate embryo sac. So, and 8 nucleate stages of the embryo sac. So there occurs 3 successive mitotic division to form an 8 nucleate embryo sac. So all these division, if you see these 3 structures, they are free nuclear. So what do you mean by 3 free nuclear? That is, only nucleus is getting divided, but there is no formation of the cell wall, right? So these divisions are free nuclear. That is, the nuclear divisions are not followed immediately by cell wall formation. So in a single cell, now actually 8 nuclei are present, right? So the divisions are free nuclear. So we saw the 2, 4 and 8 nucleated stage. So what is the next step then? After the 8 nucleate stage, that is 8 nuclei are formed now, right? The cell walls are laid down, leading to the organization of the typical female gametophyte. So how the female gametophyte is formed now? The cell walls are laid down, leading to the organization of the female gametophyte. So in the previous step, we saw that there is no wall formation. So the next step is the formation of the walls. So how they are formed? So if you see the structure here, so this is the structure of a mature embryo sac or a female gametophyte, right? So 6 of the 8 nuclei are surrounded by cell walls and organized into cells. Now the nucleus are surrounded by the cell walls. So for example, if you see here, surrounding each nuclei, there occurs a formation of the cell wall, that is separation or the differentiation, right? So here 3 cells. So 6 of the 8 nuclei, out of the 8 nuclei, 6 are surrounded by cell wall, and remaining 2 nuclei, that is the polar nuclei, are situated below the egg apparatus in the large central cell. So which is the largest cell here? This is the central cell. This is the large area and it contains the 2 nuclei, which is called the polar nuclei. And these 2 nuclei are not divided by the cell wall. There is no cell wall in between these 2 nuclei. Okay, so this is the largest cell. But what about the other cells? That is, a typical mature embryo sac is 8 nucleate and it is 7 cell. Why it is called 8 nucleate and 7 cells? So if you see the number of nucleus, it is 8, right? 1, 2, 3, here 4, 5, 6, 7, 8. So it is 8 nucleated and 7 cell. Why it is 7 cell? Because here the two new polar nuclei are, that is, they are formed together. There's no wall formation. That is, they are present in the single large central cell. Okay, so 7 cell and it is 8 nucleated. And 3 cells are grouped at the micropylar end to form the egg apparatus. So what is egg apparatus? So the questions may come about the egg apparatus, or what is the antipodal, or where the antipodals are present in which end. So we have to be clear with this diagram about each and every part of it, right? So 3 cells are grouped at the micropylar end. So this is the micropylar end, and here the 3 cells are grouped. One is the egg cell, and here these two are the synergids. So 3 cells are grouped at the micropylar end and they form the egg apparatus. So it consists of 2 synergids and then 1 egg cell. So these 3 are together. It is called the egg apparatus. This area together is called the egg apparatus, egg cell and 2 synergids. Okay. So the synergids have special cellular thickenings at the micropylar tip. It is called the filiform apparatus. So this is the filiform apparatus that is present at the micropylar end. So the main purpose of this filiform apparatus is to guide the pollen tube into the synergid. So actually the pollen tube must enter into the ovule to fuse with the egg, right? So this filiform apparatus, it helps to guide the pollen tube into the synergids, for entering into the synergid. In most of the cases, the entry of pollen tube is only through the micropylar end. So the pollen tube enters through the micropylar end and it is guided by the filiform apparatus. And 3 cells at the chalosal end. So chalosal end is opposite to the micropylar end. So this is the chalosal end, right? So these 3 cells are called antipodals. So they are called the antipodals. And the large central cell has 2 polar nuclei. So this is the large central cell. It has the 2 polar nuclei. So this is the structure of a mature embryo sac, which is also called the female gametophyte. So I hope you're clear with the structure, right? So the diagram is more important. So they may ask you about the labeling, or what is an egg apparatus. So how many cells are present and how many nuclei are present. So they will have the possibility of getting many questions regarding this. Okay, so you have to be clear with the diagram. So clear with the diagram, right? So this is the formation of the female gametophyte overall. So meiotic tetrad, that is integuments are present, and 3 of the, that is they form 4 megaspores, out of which 3 of the megaspores they degenerate and only 1 megaspore is functional, and then they undergo 3 mitotic divisions. So without the wall formation, and then this through the micropylar end, the pollen tube enters and it fuses with the egg, right? So there occurs an fertilization. So this is the structure of the mature female gametophyte. Okay, mature female gametophyte or embryo sac. So you're clear with the structure of gynoecium, right? So under gynoecium we saw about the ovule structure and then about the embryo sac. Okay, so we'll discuss few questions. So the embryo sac is monosporic when it develops from one of the 4 megaspores of the megaspore mother cell, 3 megaspores of the megaspore tetrad, 2 megaspores, the megaspore mother cell where meiosis has occurred, but cytokinesis does not take place. So why it is called as an monosporic embryo sac? So the embryo sac is monosporic when it develops from, the option is A, that is one of the 4 megaspores of the megaspore mother cell, right? So one of the 4 megaspores of the megaspore mother cell, it develops into one embryo sac. That is 4 megaspores are formed, and out of it only 1 spore it develops into one embryo sac. So it is called the monosporic development. So one of the 4 megaspores of a megaspore mother cell, and it is not the 3 megaspores, right? and it is not the 2 megaspores. So the megaspore mother cell where meiosis has occurred but cytokinesis does not take place. So this is also a wrong option. So the correct option is option A. So for the formation of embryo sac, the megaspore mother cell undergoes 2 meiotic and 2 mitotic divisions, and 1 meiotic and 3 mitotic divisions, 2 meiotic divisions, 1 meiotic and 2 mitotic divisions. So which one will be the correct option for this? That is, if an embryo sac has to be formed, the megaspore mother cell, that is the megaspore mother cell, first it has to undergo. So what will be the correct answer for this? The megaspore mother cell, first it has to undergo meiosis, right? To form megaspores. Yes, B is the right answer. So first the meiotic division has to happen, and after meiotic division, 3 mitotic division, right? So first step is the megaspore mother cell should undergo meiosis to form megaspores, and out of the 4 megaspores, only 1 megaspore is functional. So this functional megaspore, it undergoes 3 mitotic divisions, right? It undergoes 3 mitotic divisions and leads to the formation of embryo sac. So you are clear with it, right? So first to form a 2 nucleate, and then 4 nucleate, and then 8 nucleate, and it is not 2 meiotic and 2 mitotic, and it is not 2 meiotic or 1 meiotic and 2 mitotic. So B is the correct option. So as the anthers mature and dehydrate, the dash separate from each other and develop into dash. So when the anthers mature and degrade, that is dehydrate, so what will happen? What will separate and they develop into what? So here it is, the question is when the anthers mature. So what will be the correct answer? Whether it is megaspore embryo sac or microspore pollen grains, megaspores, or megaspore microspores. So are you able to follow this question? Yes, B is the right answer. So when the anthers mature and dehydrate, the microspores, first it will be present in the form of a tetrad. So the microspores, that is the microspores, they separate and they dissociate from each other. So from the tetrad, they separate to form the individual microspores and they develop into pollen grains. So B is the right answer. So megaspores, they are associated with the female reproductive part, right? So here it is asked anthers. So you see clearly and then answer. So B is the right answer. The female gametophyte or embryo sac of typical dicot, that is polygonum or monosporic embryo sac, is 7 cell, 7 nucleate, or 8 cell, 8 nucleate, 7 cell, 8 nucleate, or 8 cell and 7 nucleate. So the correct option is C is the right answer, because it is 7 celled and 8 nucleate. So be clear with the things. Sometimes maybe in a hurry and may mistook the thing. So it is 7 cell and 8 nucleate. So be clear with that, okay? The embryo sac is also known as microgametophyte, microsporangium, and megasporangium. So which is the correct answer? Embryo sac is also known as embryo sac, it is asked. Embryo sac is also known as, no, I think it is not D. That is megasporangium is also called the ovule. So it is not the D option. So what will be the correct answer? So here it is asked, embryo sac is also called So the correct option is B. Actually, it is a megagametophyte. Megasporangium is ovule. Ovule is also called megasporangium. So embryo sac is actually, what is embryo sac? That's what we saw, right? That is an 8 nucleated and a 7 cell structure that is immature to form a mature embryo sac, which is an polygonum type, and it is also called the megagametophyte or the female gametophyte. Okay, so embryo sac is called the megagametophyte or a female gametophyte, and megasporangium is ovule. So we have to be clear with it. So it is not a microgametophyte or a microsporangium. So microsporangium you will see only in the, yesterday we saw, right, in the male reproductive part, microsporangium, and then pollen grains. So pollen grain is also called the male gametophyte, and the embryo sac is the female gametophyte or megagametophyte. Okay, clear? So B is the right answer. Then egg apparatus of an embryo sac consists of egg cell only, egg cell plus 3 antipodal cell, 1 oosphere plus 2 synergids, and 1 oosphere plus 2 synergids plus 2 antipodal cells. So which is the correct option? So the egg apparatus, it consists of, option C is the right answer. That is egg plus 2 synergids, they together, it is called the egg apparatus. C is the right answer. The largest cell in an embryo sac is an egg, central cell, synergids, antipodal cells. So which is the central cell is the right answer, because this is the largest cell than the other cells. Okay, so we'll discuss few questions. So what would be the ploidy of the cells of a tetrad? Whether it is haploid, diploid, polyploid, or triploid. What would be the ploidy of the cells of a tetrad? So tetrad, actually, what is a tetrad? Whether it is haploid, diploid, polyploid, or a triploid. Yes, haploid. It is a option A. Haploid is the right answer. They are the individual cells that are formed. That's called the tetrad, right? So it is haploid. Then select the odd one. Pollen grain, antipodal cells, synergids, and egg cell. Select the odd one. Pollen grain, antipodal cells, synergids, and egg cell. So which is the odd one among these? Yes, A. That is the pollen grain is the odd one, because all the other cells are present in the embryo sac, right? So pollen grain is the odd one. A is the right answer. A multicarpillary apocarpus gynoecium is found in hibiscus, mechelia, papaver, all. So which is the correct answer? Multicarpillary apocarpus gynoecium is present in hibiscus, mechelia, papaver, and all. A multicarpillary apocarpus gynoecium. So actually the option is mechelia. Is the right answer. That is B is the right answer. Okay, multicarpillary apocarpus gynoecium. Then to some extent, viability of pollen grains depends on temperature, humidity, both A and B, and light. To some extent, viability of pollen grains, it depends on, both A and B. That is, it depends on both the temperature and humidity. So C is the right answer. Then, which part of the reproductive structure produces both enzyme and hormone? That is archisporium, middle layer, tapetum, and endothelium. So which part of the reproductive structure it produces both enzymes and hormones? Yes, it is tapetum. So we saw in detail about tapetum yesterday, right? So tapetum, it produces both enzymes and hormones. Ubish bodies are produced in embryo sac, endothecium, pollen grain, and tapetum. Ubish bodies. Yes, D is the right answer. Produced by tapetum. Then tapetum is option A, parietal in origin and is the innermost layer of anther wall. And option B, modified endothecium of anther wall. Option C, outermost layer of sporogenous tissue modification. Option D, parietal in origin and is the innermost layer of ovule wall. So which is the right option? Tapetum is parietal in origin. Yeah, it is A. That is, it is the innermost layer of anther wall and it is not the ovule wall, right? So it is associated with the anther. Then pollen grain represents female gametophyte, male gametophyte, sporophyte, anther. Yes, it is a male gametophyte pollen grain. Anther is generally composed of 1 sporangium, 2 sporangia, 3 sporangia, or 4 sporangia. Anther is generally composed of 1 sporangium, 2 sporangia, 3 sporangia, or 4. Yeah, 4 is the right answer, that it is tetrasporangiate. And occurrence of more than 4 spores in a tetrad is called polysyphony, polyspermy, polyspory, polyembryony. Occurrence of C, that is polyspory. More than 4 spores in a tetrad, that is called the polyspory, that is more spores are present. Then ubish bodies are associated with the development of embryo, pollen grains, endosperm, embryo sac. Ubish bodies are associated with the development of a pollen grain. Yesterday we saw, right? So ubish bodies, they are associated with the development of pollen grain. That is, they are associated with the formation of exine of pollen grain. That is the outer layer of the pollen grain, right? So B is the right answer. Then essential whorls of a flower are calyx and corolla, corolla and gynoecium, androecium and gynoecium, all of the above. Essential whorls of a flowers are calyx and corolla, corolla and gynoecium, androecium and gynoecium. Yes, C is the right option. Its essential whorls are androecium and gynoecium because they are the structures which are involved in the reproductive part, right? And the other calyx and corolla are the accessory whorls. Sporopollenin is found in exine, intine, cytoplasm and nucleus. Sporopollenin is found in exine, intine, cytoplasm and nucleus. Yes, A is the right answer. Sporopollenin is found in exine. Okay. Then microsporophyll of angiosperms is known as androecium, anther, filament and stamen. Microsporophyll of angiosperms is known as androecium, anther, filament, stamen. So, which is the right answer? Microsporophyll, stamen. D is the right option. Stamen is called the microsporophyll. So stamen. So from stamen, they have the anther and the filament. So stamen is only called the microsporophyll. Okay. So D is the right answer. And main function of endothecium in anther is mechanical, nutritive, dehiscence, and storage. So the main function of endothecium in anther is, yeah, B is the right answer. That is, it is involved in dehiscence. See that is mechanical, nutritive, dehiscence and storage. Dehiscence is the right answer. The endothecium is involved in dehiscence. Okay, it is involved in the dehiscence of anther. Is the right answer. Which one is the female gametophyte? Embryo, embryo sac, endosperm, and pistil. Which one is the female gametophyte? Embryo, embryo sac, endosperm, and pistil. B is the right answer. That is embryo sac. Embryo sac is also called the B, female gametophyte. So you have to be clear with these terms. Okay. Embryo sac is the female gametophyte. So ovule is also called megasporangium. So these terms we have to be clear because they will ask some questions based on these terms. Embryo sac, stamen, anther, and what is a male gametophyte, female gametophyte, microsporophyll, then megasporophyll. So these, so megasporophyll is carpel. Carpel is called the megasporophyll. So we have to be clear with these terms. Okay. And pollen grain is the male gametophyte. Then grassy nucellate ovule shows absence of nucellus, well-developed nucellus, partially developed nucellus, and modified nucellus. So which is the right option? Grassy nucellate. So we saw right about the terms tenue nucellate and grassy nucellate. So grassy nucellate ovule, it shows B is the right answer. It shows well-developed nucellus. Shows well-developed nucellus. Okay. Then perisperm is persistent nucellus in seed, ovule wall, ovule coat, and fossil of Hastoria. So what is the right answer? Perisperm is persistent nucellus in seed, ovule wall, ovule coat, and fossil of Hastoria. So what is perisperm? So it is the correct option. So A is the right answer, right? Perisperm is persistent nucellus in seed. That is, even after the formation of the, that is, ovule will form into a seed, and even after the formation of that seed, if nucellus is present, that is called the perisperm. So are you clear, Nandini? Are you able to follow? Okay. So Filiform apparatus are found in antipodal cell, egg cell, secondary nucleus, and synergids. Filiform apparatus are found in, D is the right answer, that is synergids. Okay. Megasporophyll is called stamen, carpel, ovary, stigma. Megasporophyll is called stamen, carpel, ovary, and stigma. B, carpel is the right answer. Then Amphitropus type of ovule is whether it is straight, inverted, transverse, or coiled. Amphitropus type of ovule is whether it is a straight ovule, inverted ovule, transverse ovule, or coiled. B is the right answer. That is, Amphitropus ovule is an inverted ovule, right? Amphitropus ovule is an inverted ovule. So actually this Amphitropus ovule is present in most of the angiosperms, right? So it is an inverted ovule. Then horizontally oriented ovule in angiosperm is called Amphitropus, Hemitropus, Camphylotrophus, and Trophus. So what is the right option here? So again, B is the right answer. That is, Hemitropus ovule is also called horizontal. Hemitropus ovule is also called horizontal. Okay. And which of the following types of embryo sac is mostly found in angiosperms? Whether it is bisporic polygonum type, tetrasporic polygonum type, monosporic organ type, and monosporic polygonum type. So what is the correct answer? Which of the following type of embryo sac is mostly found in angiosperms? Yeah, it is an monosporic polygonum type, right? Monosporic because it is formed from a single megaspore. Then when hilum, chalaza, and micropyle lie in one straight line, then that ovule is called Amphitropus, Orthotropus, Camphylotrophus, Anatrophus. When they all are lying in a straight line, it is called the option B is Orthotrophus ovule. All hilum, chalaza, and micropyle is in the straight line. It is Orthotrophus ovule. Then in angiosperms, all the 4 microspores of tetrad are covered by a layer which is made up of pectocellulose, callose, cellulose, and sporopollenin. So which is the right answer? In angiosperms, all the 4 microspores of tetrad are covered by a layer which is made up of callose. So B is the right answer, right? So B is the right answer here. Made up of callose. Then embryo sac is represented by megagametophyte. Yeah, this actually is all right. Megagametophyte, megasporophyll, microgametes, and megaspore. So the correct answer is megagametophyte. Embryo sac is represented by megagametophyte. What does angiosperm mean? Naked seed, covered seed, leaf fall, seed bud. So which is the correct option here? What does angiosperm mean? Angiosperm, naked seed, covered seed, leaf fall, seed bud. It's not A but B. Covered seed, naked seeds or gymnosperms, right? Only gymnosperms or naked seeds. So here in angiosperms they are covered seeds. So B is the right answer. Are you clear? So gymnosperms, they have the naked seeds. Okay, so here they are flowering plants. So they are covered seeds. Okay. So with this, we have completed the pre-fertilization structure and events, right? So under that, we saw in detail about the androecium and gynoecium. So tomorrow we'll see about the pollination, that is about the different types of pollination and about the pollinating agents. So you go through this too, that is about androecium and gynoecium, and practice some questions related to it. Okay. So any doubts? Okay, then thank you.